So here's a study, one of the most important, isolation and rapid sharing of the 2019 novel coronavirus SARS-CoV-2 from the first patient diagnosed with COVID in Australia. This was the first non-Chinese isolation study done. Very important study. Same thing, viral culture material from initial nasopharyngeal swab was used to inoculate a viro-slam cell line. So you know what's happening now. You know what they did. Now here's the most important thing. And I'm going to read it, and this will not make sense, and then I'm going to explain it. Quote, electron micrographs of sectioned viro cells showed cytoplasmic membrane-bound vesicles containing coronavirus particles, see box 5b. Following several failures to recover virions with the characteristic fringe of surface spike proteins, it was found that adding trypsin to the cell culture medium immediately improved virion morphology. Let me explain that. So he did this whole thing that I just explained, and he found these particles, right? And he shows them on his electron microscope picture. Mind you, there's lots of other different particles in this slide. But he points an arrow and he says, those are the SARS-CoV-2, because I can see this characteristic spike protein. You've all seen that, right? But then he says, we didn't get them. They didn't look like the spike protein. So we added trypsin, which is a protein digesting enzyme, and then we could see the spike proteins. In other words, you have this thing which has a protein coat, and so you only see the spiky things if you dissolve the coat. Or, as I said, you want to get a box of wood and you look for star-shaped pieces of wood. But you look and they're all square. So you call your carpenter friend and he cuts out the edges and look, stars. Or as Andy said, you go looking and you see a pig and you stick some feathers on it and look, a chicken. In other words, you don't see the spike protein unless you add trypsin. There is no study that I've seen, and believe me, because I was under the gun on this, I've looked, they all have to add trypsin. Now, here's where it gets really wonky, because then he says, which confused this Australian lawyer, so then we do a genome analysis. They said, we found that the results of this culture, we did a next generation genome sequencing, and we found a 99% homology match with the original Wuhan genome. In other words, the genome that they found in this was 99% similar to the published genome of the Wuhan virus. That clinches the deal. They found the Wuhan virus. It has approximately 5 billion pieces of RNA and DNA in it. Then they said, okay, we're looking for something. They call this the unbiased de novo next generation sequencing. Unbiased means they're not gaming the system. De novo means anew. They're going to find the genome anew. Next generation is just the name for the new way they find the virus, the sequence of the genome. So they start with 5 billion pieces of RNA and DNA. You would think they would look through this brew and find the genome, right? They don't do that. The first thing they do is they discard the DNA, and that's called unbiased. And then, so now they go from 5 billion pieces of DNA to 56 million, approximately. And then they discard all the pieces of RNA. So now you just have RNA that match humans or other microbes, like bacteria or fungus, based on published databases. Now, mind you, we already know that you can't tell whether a RNA is from a virus from the outside or the breaking down of the U. In fact, we now know that the spike protein sequence has been found 93 different times in the human genome. We know that. But anyways, they threw out all the ones that were published to be part of the DNA of humans or other microbes of the RNA. So now we have 23 million, and that's called an mRNA library. Now, the best way to think of this, I hope you're still with me, is so they started with 5 billion letters and short phrases. And they threw out, I would say, arbitrarily 90% of them. And so now they have 23 million letters, words, or short phrases. You with me? And they say to you, align this into a book. You might want to ask the person, which book? Right? You can't take 23 million letters and arrange them and come out with a book. Maybe you could, a computer maybe could, but they can't. So here's the key to the kingdom. They have to put in a template and say, take these 23 million letters, take these 23 million little sequences of RNA, and organize them into Hamlet. Or in this case, they put in SARS-CoV-2, the sequence from China. Why did they put that sequence in? Why not measles virus sequence? Because the guy had COVID, and because we found the spike proteins after we put in trypsin. And so we know it was SARS-CoV-2. So we put the genome in, and we say to the computer, match it to that genome, and it does, 99%. And then it makes a few mistakes, and those are the variants.
But they could put measles in there.
They could put measles in there, and they would get measles. And they could put HIV in there and get HIV. And they could put Zika in there. And there's enough words and letters and phrases to make any sequence you want. It's all about which template you put in. Because if you put Hamlet in, you can arrange those 23 million letters to make Hamlet. If you put War and Peace, I think, except if you put War and Peace, you might miss a few. And so then you have to add them to the end, which is what they call sophisticated alignment processing. They just add them when the gaps are. And that's the genome. And that's called the complete genome. And that's nuts. And the variants are because, a computer missed a few of them. And anyways, this guy doesn't have the same letters to make the sequence exactly the same. So they fudge it a little bit and get different parts are not 99, they're 98% aligned. And that becomes the variant. Has everybody got it? No, anybody doesn't? Yeah.
When does the study come out?
What? From the first guy who did it in Wuhan. And he got the template from the third guy, and he got it from the second guy, and he got it from the first guy, and he got it because he matched it with SARS-CoV-1 from 2003, which was done exactly the same way, and he got it from matching it with MERS virus in 1997, and this is called in silico genome sequencing. In other words, it only lives in the computer. There is no There is no complete genome in the brew. Cannot be found. 100%. All the virologists agree. You have to align it. You have to cut it into little pieces. Like if you had just phrases, you might not be able to make War and Peace. So you cut it into letters, and then you can make War and Peace. So they cut it into letters, throw out the parts that are French, because we don't want War and Peace in French, and then they use the template, and we're going to show that by putting RNA from yeast and blindly sending it and saying, is this a sample of measles virus? They're going to say it's a sample of measles, Zika, HIV, SARS-CoV-1, and that should be the end of the story.